And I'm also going to, let me see if I can share some other way. There's an AI meeting notes app. Oops. Lots of different buttons. Well, welcome. Today I'm going to be talking about compost tea for the organic grower. Thank you all for being here. Thank you to those who are just curious. I would love to know in the chat if you'd like to put down where what your questions are about compost tea. I would love to know it. This has been on my mind for like 2 years now, so I have a lot of things to say about it, and I better not waste time. But since we are a quiet group today, I'm just going to go ahead and introduce myself. So I'm Nikki Schauder. I compost in 3 ways. I research and use compost tea for food production, but I've always been interested in this concept of compost tea ever since, well, before I forget, ever since I have started composting and learned about that you can make compost tea. Apparently, we're brewing it in order to make more bacteria, good bacteria, grow in a brew and spread that in our gardens. And that just really intrigued me. So I went to Southern Sustainable Agricultural Research Education. I hope everybody can hear me. And I'm not just recording nothing, a bunch of slides. I'm going to put up my own volume here. So I wanted to, so Southern SARE has made this research possible. Sustainable Agricultural Research Education has a mission to create sustainable, profitable economies, especially locally for small farmers around the United States. So any farmer or producer, in fact, you don't even have to be a farmer, can apply for a competitive grant with Sustainable SARE, and you can study something that could help benefit farmers. And I definitely wanted to do that since I'm now in a farming region. And I did particularly, I had composted in various ways. I see people are in the chat. Hi, I'm joining from my work, so microphone is off. That's great, Susan. And your question is bokashi composting. What should it look like when it's ready to mix into soil? And can bokashi composting be used for compost tea? That is a great question. And actually, I think the researcher that I'm collaborating with has a blog on that in particular. So that is a great question. These kinds of questions are what come up, right? People tell us, oh, use this. This is a great indoor way of composting. And then when you're done, it has a different, distinct smell, very fermented smell. And this is the bokashi. So this is how I've composted in the past in our townhouse. I still have these bins, but I don't use them anymore. Not because I don't want to, but because the composting is already serviced by a large composting bin that we have outdoors. And this is great if you live in a small area. How do you know when it's done? So I believe there are instructions. When we bought this particular bin, it came with videos. And this is to answer Susan's question about when you know it's done. I think it was like 4 weeks into the process. It doesn't look much changed, quite honestly, except that it does smell fermented. So just think of it as a ferment, a pre-ferment to becoming compost. It's not finished compost when it comes out of the bucket. You still have to put it out in your garden, and then it decomposes some more, but it has a different composition of microbiota that is more lactobacillus in there. So is that good or bad? That's one of the reasons that I wanted to do this research on compost tea is, well, what exactly am I brewing in my compost tea bin? So here's a survey, a little preview of the research area that I used in my backyard, formerly a parking lot. So this is my research beds. I have 9 of them. It is April 26th, 2024, and this has happened. These were planted last in, let's say, February, March, transplanted, that is, and in a few months, here's the results of this.
some of these beds have already been harvested, so I'll have to account for that. But it's not lost upon me how fortunate, how privileged I am that this is my daily job, that I get to do this every day and look at the plants and see how they change and study them. So I'm going to go ahead and take measurements. This is a control. This is the untreated, nitrogen treated bed, and it has been harvested the most because these were, believe it or not, these were huge. And this is the compost bed. Beyond there, so each of these rows have different N treated, control, and compost treated. So this would be a compost bed right here. This is a compost bed. This is a control, and that is a N treated. This is a control, a compost bed, and an N treated. And from viewing this, it's not quite sure. You can see.
Oops, I'm sorry, that went backwards. But what I went to say was you're not quite sure what the results are. Like it seems different. And studying it further, you will look at the results later on. But studying it further, the results do need further research and more than 9 beds to be statistically significant. But along the way, I learned so much that I can't even share half. Like I can probably share like 3/4 of it. I'm trying to share as much as I can here, but so many little details about composting, compost tea, and the different kinds of compost. So here's how I thought it would be best to present the information that I do have with you all in the hour that we have. First, to just clarify what this term composting means, because when I started out, different researchers were saying different things and had different terminology. Different farmers would use compost tea to mean different things. The second is, what exactly was I testing? Why would one consider using composting in the first place? Is it really helpful or is it not? Why bother? And what it, after determining what it is. And then the third is just how exactly I measured all the things that I measured and who I collaborated with, because I'm not a scientist, I'm just a farmer, and certainly some of the things that I did were more farmer-like than scientist-like. But just to throw it out there that you could do this at home. And then the fourth is, if you were to do compost tea, the many ways that you could do it and the many foundations for compost tea that you could use, so different composts for different teas. And you're welcome, Susan. So if you have any questions, drop it in the chat, and I will try to answer as much as I can. So the first thing that came as a Google search, because 2 years ago I wasn't using ChatGPT, I was using Google, was that compost tea provides no real benefits. That was the #1 search that came up, and I was like, this can't be right. And that's what made me want to explore this even more, because I was definitely on the camp of like, there must be something good about compost tea. Otherwise, why would we be using it, or why would people be even using it, or farmers? But the two words compost tea, like I mentioned earlier, came to mean different things. Historically, here in Native American indigenous tribes used plant-based extracts for soil enrichment and pest control. So here again, we think, oh, it's for fertility and pest control. And these teas were made by steeping plant material in water, often including plants with known pest repellent or nutrient boosting properties, such as stinging nettles or other nutrient-rich herbs. Fish-based fertilizers, like burying fish remains near crops, were often combined with organic matter, creating conditions like liquid compost tea, but in the soil. So there's some practices already in place here in America, at least, that were kind of alluding to the compost tea as we know it now. But it wasn't until Dr. Elaine Ingham, who is an American microbiologist, did a lot of work, and she was working with the USDA and created a manual on how to properly, safely brew compost tea, that this became more of a trend in the regenerative, sustainable, organic farming sectors in America and the world, really, because she was the first to research it using the microscope at that time with the technologies that she had 20 years ago. And she had proven that compost tea application, specifically in warm weather climates, which was most of her growing was in California and in Africa, they really worked well. And so she has grown a lot of she has grown this like soil food web organization with several students who have studied under microbiology and microscopy under her to to say that yes, this does work. You can increase the number of microbiome microbes, so bacteria, fungi, nematodes in your compost from your initial compost tea by brewing it. And so that was her main studies 20 years ago. And since then, a lot of people, but first she was also, because of that, she was also the first one to qualify what exactly compost tea was. In particular, in this study, I am using aerated compost tea, which is bubbled compost tea, not just steeped like we would normally make compost tea at home. I mean, we would normally make tea at home with a tea bag steeped into a mug of water, right? But in the compost, as she started teaching, she and her students started really prescribing or more defining the different kinds of compost teas. Now, this is all good, fine and dandy if you're studying under Dr. Elaine and you're in the same circles, but as you go out and you're no longer in those circles that Dr. Elaine inhabits, you will find people using arbitrarily compost tea and anything that's steeped in any compost steeped in water as compost tea, which is, I think, is fine. But for the purposes of this research, just wanted to qualify, and especially if anybody here is coming from her camp, that the compost that I made was aerated compost tea, which is bubbled compost. Later on, they found that they had to be specific about whether you fed the aerated or the brewed compost tea some sort of a feed like molasses or fish hydrolysate in the process. And if you did, then you called that an activated aerated compost tea. And if you didn't, then it didn't work. Then it was not compost tea. So she and her students will tell you or recommend that if you only bubble compost tea and you don't feed it, then basically it's an extraction and there's no life in the tea. And so that was my first introduction to this, as I was just starting off. Am I making a good tea to be good for this research? Is coming up against a lot of this. This must be done in this way or it doesn't work. And so this added more questions to my research, because I'm like, well, why wouldn't it work? What exactly am I growing in the compost water that I'm brewing? The last here that I'll talk about is compost leachate, which are the yucky dregs of compost that pools and is likely anaerobic. Now, when I was first introduced into the topic of compost tea, very many people, especially those who studied under Dr. Elaine, would say that is the worst. Those things, you never know if they're good. And those leachates, just similar to what would come out of a bokashi bin, it's like this gooey stuff, anaerobic for sure, because bokashi is anaerobic, right? But why does the bokashi instruction different than this instruction, for instance, that says you shouldn't use it, the instruction of Dr. Elaine? So I could see like conflicting messages here, whether compost leachate was good or bad. And so I just want to clarify this from the get-go. A bunch of Koreans, scientists, eventually, because of their background in Korean natural farming and their indigenous practices that involved fermentations of plants and animal materials into liquid fertilizer, very similar to bokashi. Bokashi sounds very Asian, right? It's a Japanese version of this. And so they picked up on Dr. Elaine's research and riffed off of it, looking at her studies, and they wanted to see what the effect of aerated compost tea, notice that they used her language, aerated compost tea on the growth, specifically of lettuce, soybean, and sweet corn in organic cultivation. And so they opened up, and for me, like reading this research was pretty eye-opening, because instead of simply using one prescribed way of composting, which in Dr. Elaine's case, she went by the USDA as a specific way that you need to make your compost so that it is pathogen free, because that's very important for farmers, that you make sure that everything is pathogen free, that you have to heat it up and turn it a certain number of times until it gets to 105 or 112, I can't remember anymore, the temperature for 3 days, and then you know that you've killed everything, and then it becomes finished compost. So it was a thermophilic compost that she used as a base for her compost teas. And other studies did the same, but this particular research looked at 3 different kinds of compost. So they tested it on vermicompost, which is compost from the Eisenia fetida worm, a red wriggler worm, oriental medicinal herbs, hinoki cypress bark compost, because Korean natural farming has a lot of different composts that they can choose from, and rice straw compost, 2-year-old composted rice straw. Again, using rice as the fermenting agent, I believe, in this case. So what they did was, what they wanted to know was, why use composting in the first place? Does it make these soybeans, lettuce grow bigger? And that was my question as well. So in the course of my research, I wanted to answer 3 questions. One was, does it indeed multiply the population of bacteria in your compost tea? Does the compost tea that you start out with, is that less than the amount of bacteria that you end up with after brewing or treatment or feeding? So that was one of my questions. The second was, does that compost tea have more nutrients than after you brew it and treat it than what you initially started out with? So is it worth brewing the compost tea in order to get those nutrients? And then the third was, can compost tea be a viable alternative to conventional organic inputs that farmers use, so that they can lower their cost of growing their operations and have a small input for a big yield, right? And in permaculture, what's even wonderful about that, we bring back all that compost and put it back again, which was, I thought, was the beauty of this research was, and then we could make it tea again, and then you could just bring it back, and it's a good cycle that just keeps on getting better and better in our gardens. So we can do this with compost. Maybe we could do this even better with compost tea. And that was my question. Is it better with compost tea? So back to the Korean study, they looked at 4 different compost combinations. So one was just vermicompost, which is this VC white box. One was the Oriental Medicinal Herbs compost, the Hinoki cypress bark compost, which is this gray box. Rice straw compost, which is the blue dotted box. I hope I'm recording, yes. And then a mixture of everything, which is the darkest box here, or darkest line, or darkest column. So they aerated this compost tea. Now, at least in the research that I saw, they didn't mention in their paper what exactly they fed it. I think they just assumed that they fed it something, or maybe they didn't. But I couldn't find, so I tried contacting the researchers, but I didn't get a reply. So I didn't find what they were feeding the compost when they were brewing it. But by brewing, I mean that they aerate it, they bubble a bucket of water with these composts added to them for 1 day, 2 days, and 3 days after brewing. And then you see the different results. So I want you all to take a look at this, this graph. In the next graph, it's the same, and answer the question, which type of compost performed the best, do you think? I'll sip some water while you guys think about that. Which type of compost performed the best? And you could drop it in the chat if you guys have any. Yeah. The combination, MOVR, yep. Yep, Andrea, thank you, and Susan. So, what do you think um, that kind of tells us about the contents of our compost? What's the most effective kind of compost, do you think, just based on this initial research? Diverse inputs. Yeah, so the more diverse compost you have, the more diverse your compost is, the more effective it is. And I have diversity. Yeah, hi Miriam. Yes, so the more diverse your compost is, the more diverse the results will be. But here, they used the best performing compost, which was the MOVR or the compost mixture, to see the growth of a soybean plant, and they found that at 0.4 and 0.8, hardly any difference really, percent, it grew very well. So my first research wanted to ask the question specifically, does aerated compost tea work on leafy greens specifically? My second research was on tomatoes. and both research, and this particular research, out of this research, I stumbled on another thing that came up, which was initially I didn't want to look at them. I didn't really think to look at what the microbiota was of a compost tea, but this research had it in it, and it just gave interesting results. So I'll talk about that in a little bit, but here's the research purposes of ACT, or aerated compost tea, on leafy greens. One, is ACT as effective as their commercial counterpart? Does ACT improve germination? So I wanted to see, is it good for me to soak my seeds in ACT water rather than just watering them in plain water? And then the third is, does ACT increase nutrient density? And for that, I would send out, and I have sent out the leafy greens to a lab, and they've analyzed each of my leafy greens in nitrogen, in control, and in ACT. They've analyzed their nutrient quantities in each of them. So how exactly did I do that? So I'll start with the compost tea. How I brewed the compost tea was in these buckets, very simple. I wanted this research to be as accessible to anyone as possible, from the gardener or small farm to, anybody. So you use a fish pump, you aerate the compost tea, and then you bubble it for, my initial research was 24 hours. And after that, I take a sample of it. I sent a sample to the lab. And after that, this bucket was poured into the beds that were designated ACT, or compost tea beds, in those 9-bed plots that I showed you earlier. Now, you might be asking, why does compost tea need to be bubbled? And that was my questions particularly. So according to Dr. Elaine Ingham, compost and compost tea need to be produced in oxygen-rich aerobic environments. This ensures that the finished product is rich in nutrients and beneficial organisms. When compost or compost tea is made in anaerobic conditions, nutrients are lost to the atmosphere, which is why anaerobic compost stinks. The end product of anaerobic decomposition may also contain alcohol, phenols, organic acids, and proteins that are toxic to plant tissues. Now, since saying this, which is, I believe, about 10 years ago or more, we have seen the advent of DNA technology, DNA analysis, even in the fields of regenerative agriculture, and looking at the different bacteria that we have. What I think, for Dr. Ingham's time, the prevailing view was aerobic is the favored bacteria for everything, because a lot of the pathogenic bacteria lie here in the anaerobic. So these are the enterobacters and the Clostridia. And again, now we're realizing that it's not all black and white. And that there are some anaerobic bacteria, such as the bacteria in bokashi, the lactobacillus bacteria in ferments, and even more like Aeromonas. I got a lot of that anaerobic bacteria. That was the end bacteria of my compost bin was a lot of Aeromonas. And they're facultative. So they're facultative. There's aerobic, to back up. There's aerobic, which are air-loving bacteria. There's anaerobic, which don't need a lot of air, so they are low, or some generally anaerobic. And then we have facultative, or bacteria that straddle the two spheres. So they can switch according to where they are. So if they happen to be in an aerobic environment, they can still thrive. And if they happen to be in an anaerobic environment, they'll just adjust a little bit and they can still be alive. And a lot of the enterobacters and the pathogens are in the anaerobic. And that's why I think, the general prevailing view was don't brew, you need to brew and keep it aerobic. So nutrients are lost in bokashi. I don't know, because my particular study was not on bokashi, it was on vermicompost. And I decided to go with vermicompost only, unlike the Korean study, which had the whole mix of compost, because I wanted to be as simple, accessible as possible. And vermicompost studies showed that the worms that processed organic matter actually were sort of a filter to pathogens already. So they were kind of a natural filter. So I could somewhat be assured, of course not 100%, that if they were finished in a vermicompost bin, if my compost was finished in a vermicompost bin, I had a very low pathogen load. And I also tested it. So I tested it in both Elaine Ingham's lab as well as in a DNA lab to make sure that the compost tea I was starting out with and the vermicompost I was starting with had no or very insignificant numbers of pathogens. And that was indeed what I saw. So in bokashi, I don't know about the nutrient, to answer that question. The nutrient analysis of that, you would have to send out a bokashi sample of something grown in a bokashi fertile soil media, and then analyze the nutrients in that plant. So I'm going to go into that plant sap analysis in a little bit. So here we're brewing our compost tea, but for how long? So according to Dr. Ingham, ACT should be brewed for 24 to 36 hours to allow sufficient time for aerobic microorganisms to multiply to beneficial levels. The brew should be used immediately after brewing to ensure highest microbial activity. So this is Elaine Ingham's slide of the oxygen in a brew of compost tea and the activity of the microbiome. So and the one in the bottom, so here we see the activity in UG per mil of the bacteria. So that's the population of the bacteria. And then on the bottom x-axis, we see the number of hours of brewing. So at what number of hours, according to this slide, do we see the highest activity in bacteria? All right, 47 hours, Andrea says. Oops, let me go check that out. Actually, here in this particular slide, I think it falls in, so 47 is the highest number of hours that they monitored this. 18, Susan says, yeah, they're 18, 19, right? I was looking at the red. Yep. Yeah. So the red is the oxygen. So the highest amount of oxygen. So they have, what's interesting is like when we have the most amount of bacterial activity, we have a lessening of oxygen. So they're like competing for the oxygen, right? And then they brewed it, which is they're bubbling this all throughout the 47 hours. But good, thank you for your answers. Yes, so that's where she said 24 to 36 hours is optimal. Dr. Ingham says, even though her slide says about 19. In the Korean study, we saw changes of microbial population density of aerated compost tea at room temperature for 4 days after brewing the air pump. So they had so this is not activity, but density of population in these slides. So we see this on day 3. Oh, 4 days. So they did it for 4 days, and we see the biggest spike on day 3. So 36 goes with what Dr. Ingham said, right? So, and this is what's, okay, I don't know if this was sent to me, if you can see anything moving in there. So I sent my samples to a Dr. Ingham lab that was looking for life in my compost tea, and there was not that much life. by the time that it got to the lab in North Carolina, and they put it under a microscope to record it, there was no more life in my compost tea. And so I also did a DNA analysis where I would take a sample, put it of my compost tea and my compost. So this was for like the compost is this container, and this was on the left was my compost tea. I get a syringe and put it in here, and it would sort of flash freeze whatever these solutions in here would freeze frame all the players in my compost tea and in my vermicompost. And that freeze frame showed me the true population and who all those players were, as opposed to the microscopic analysis where you have to go drop by drop and sort of scan each pane. And maybe you miss a drop or maybe you miss a player, right? Here, you have everybody in one drop. That's you definitely you capture that and it's sent to the lab, and then they send you back the results. So at this point, I'm gonna share a different screen. I hope I haven't lost anybody in the weeds here, because, oh, where is my screen? There we go. All right. Chronobacteria. So they have what's called now a chronograph. Okay, everyone see that? Yes, I hope so. Let me know if not. Tell me in the chat. So this is a chronograph, and the scientist who works with me is Dr. Zach Jones. He is in Colorado, and he was the one who analyzed the bacteria of the vermicompost. So, and the compost tea. So when I sent him the vermicompost that the compost tea was made of, what he did is have his partner displayed it in such a way that each color shade was a different bacterial family. So here, for instance, we say Clostridia, 1%. I have 2% Bacilliaceae, and so on and so forth. Each of these are families and orders, and you can see that I have a rainbow of different players in my bacteria. And then it's labeled here on the right side what kind of bacteria each one of those are and what percent they make up of that drop of vermicompost or like spoonful of vermicompost I sent him. Now, fast forward 24 hours later, I took another sample and flash froze it in that same vial, sent them to him at the same time. And what do we see? What's the difference between a compost tea makeup versus a compost makeup? So this is the vermicompost, and this is the compost tea. That's the vermicompost. So again, each of these are different bacterial players. Here are like the bigger families: Actinobacteriota, Proteobacteria, Chloroflexi, Bacteriodota. So that's vermicompost, and this is compost tea. And in the compost tea, we have Proteobacteria and then Bacteriodota. And then some subfamilies here. We have Pseudomonas, Enterobacterialis. All right, I see some answers. The first one is farmer diverse. So the first one is the vermicompost, and the second one is the compost tea. So what does that tell us about compost tea after 24 hours? So we definitely have a reduction in diversity. I think that's what you all had said, right? We have approve. You have a reduction in diversity from vermicompost, which is super diverse, at least my vermicompost that I sent. And then all of a sudden, all those families die, and we have an increase still of players, but only of a select few families. So we have Pseudomonas, Aeromonas, and I don't know what the third one was, and somehow some like mixture of something or another. Oh, Actinobacter, Aeromonas, and Pseudomonas here. Bubble it with worms as filters. Bubble it with worms as filters. Oh, because we have Enterobacter here as well. Yeah, so because we're not sure, right? You know, in the end, the microbiologist I was working with, he said like not all Enterobacter, like the amount of it wasn't all pathogenic. So he like deemed my in the paperwork that he gave me, he said that it was negligible, because this particular species of Enterobacter wasn't known to be pathogenic, if that makes sense. So, but I don't know. I'm not a scientist, like I said. But that was really interesting for me. And that led me and the biologist that I was working with to like think, well, what are we doing here? Are we selecting for a few families? And so that was kind of eye-opening for that first research, and asked, and because of that, I started asking questions like, well, what happens at 12 hours? What happens at 24 hours? What happens if we feed them different things? So what we do know so far is if it's warm, your biology will be more active and will spike sooner. So if you are making compost tea, it could be 6 hours, 12 hours, 24 hours on a cold day. If it's cold, it might take your microbes longer to reach their peak state. But I think because of my second research, I kind of know that the most diverse, if you're looking for diversity of bacteria, the most diverse is the beginning. So just dump your compost in water and then just pour it out. And I think if you're looking for diversity in your garden, that's the most practical way. Like the brewing of it is only for selection. So does it need to be aerobic? Conclusion. So ACT in particular is particularly effective when freshly applied, providing a surge of a few families of mostly air-loving and more pathogen-free beneficial bacteria. However, non-aerated teas or compost extracts might be more suitable for applications where a slower release of more diverse microbial benefits is desired over time. So if your aim is to increase the diversity of bacteria, fungi, and whatnot in your good bacteria that's coming from your compost in your garden, I would just make a solution of your compost and water your garden with that. And then if you are looking for disease prevention, because that's what I wanted to, you're looking for more. These are more questions that I haven't really quite answered yet. That's trying to be answered in the second research is, what exactly are we selecting for? Is it the same all throughout? When I brew my compost tea and I only end up with 3 families, because across the board for other people, the microbiologist I'm working with specifically works with compost teas. So vermicompost and compost tea brewers around the United States and even in Europe send him samples, and he sees this trend that they only get dwindled down to like 3 or 2 families. And more often than not, Pseudomonas is always in that family. So why? Why Pseudomonas? Why do we want to select for a specific family of bacteria? And does this family do something good to the plants? So I think if you know Pseudomonas, and I've heard from another talk of a wine grower, organic wine grower talk, oh, Pseudomonas is really helpful in preventing, like, a big thing with vineyards is the rot, certain molds or mildews, then compost tea brewed is the solution for fighting that rot. So you want to brew the compost tea for that specific pathogen, to go against the pathogen of the vineyard, not human pathogen. But yeah. After the tea is drained off, is the compost still diverse? Would you put more water on that compost or just use it by adding to soil? Oh, after the tea is drained off? Oh, you mean from the bag, Andrea? I just add that to the soil. I just add it all together. Like, okay, I've watered it with a compost tea. I've tried to make sure the leaves are drenched with a compost tea. Right now, our raspberries are dying from a mold, from botrytis, of which is a Pseudomonas. Pseudomonas has been shown to fight that off. And so I was telling Dave, it's because we didn't put the compost tea on the raspberries. So now they're dying. I know it's also the end of season and whatnot, but that we should have done that, drenched that on their leaves. And yeah, you can just add the compost to your garden. Good question. So for the first research, is ACT equal to or greater or better than a nitrogen fertilizer? That was my one question. Does slower release of microbes mean you can act now in fall and it will still be effective in spring? That is a great question, Miriam. There's some people who know this more than me because I have not done that research, but that is a great question to ask too. Do they persist, right? Do they persist? I think this is why some of these applications are best done in the spring, because whatever the case may be, the temperature dropping will definitely make bacteria act slower, reproduce slower. But if you want to seed them or keep that engine running, like decomposing things in the soil, you want some sort of an activity still to happen in the fall to winter months when everything is dormant, and you still need things to break down. They break down slowly, but with compost tea or compost, they could break down faster because there will be more microbes to do it. Now, will you still need a fertility treatment in the spring? I would say likely yes. You'd still need some sort of a fertility boost in the spring, but there's definitely a benefit to having done it year round, to having added compost to your garden year round. I'm planting greens now. Oh, okay. So for leafy greens, this is why I use nitrogen as the conventional practice. For leafy greens, nitrogen is the one nutrient that really helps produce leafy greens, which is your main product. It's not like tomatoes where we don't want the leaves, we want the fruit, right? So I wanted to see, was ACT a better fertilizer than nitrogen? And does ACT make leafy greens go bigger? Does ACT make leafy greens grow better? And so we started with germination. So we germinated N treated chard, kale lettuce, and then control treated chard, kale lettuce, and ACT chard, kale lettuce, and created a germination test from that. And from the tests, we found that the best germination media was still water, and that those germinated in nitrogen, those just germinated in ACT, which became stinky after a week in the paper towel, did not germinate well. So that was like, I'm coming from like, I want compost tea to win, but it didn't win in that scenario. It's just germinated in water and soil, and I think that'll be enough. I know some people will say other things, but at least that's what happened in my germination tests on paper towel. This was how I laid out the beds. For those of you who came in later and didn't see the 9 beds that I have in our property, we had a control, compost control, just a random mixture of these 3 treatments, and 20 of each of the of Swiss chard, kale, and lettuce were planted in each of the beds. And then I harvested these, weighed them, and some of them, the kale and the lettuce were weighed well, but the Swiss chard was kind of like, it was something happened in that weighing that I just have the averages of them, but not the whole, each of the 20. I don't have each of the 20's weights. So nutrient density scores. So now how can I sent each of those crops to the lab, each of those treatments of the crops to the lab, that is a bionutrient lab, to find out what their nutrients, to find out, hoping what their nutrient score, I could find some sort of a nutrient score for them. But what they did was they sent me back a lot of information, which is this. This is actually my chart, but what they sent me in particular was, okay, we have the sugars, the proteins, the phenols, the organic acids, and all of these key nutrients and all of these toxins in your plant. And I had to figure out what each of these numbers meant and which was the most important key nutrient, what were the important key nutrients, what were the detrimental toxins. So in the course of this, there's nothing really right now. They're still developing. So I was working with the lab that I was in conversation with them, and I was trying to say, is this okay? I made something like a score where I tried to score each of my plants according to nutrient density. And how did I do that? I took what they gave me as a score and their ideal for that score. So for instance, they have targets for each of these leafy greens. So the desired target that they want for chard, for instance, was the total sugar should be 16. But if my treated chard was 5.68, then I only had 36% of that. So from the long and short of it is, this was probably the bulk of my thought work went into like, how am I going to analyze all this data and put it into a score? So I went back and forth with them and told them, is this okay? Is this a good score to use? This is how I did it. This is how, and so they said, yeah, it's a good start, because I didn't weight them according to, this is a better, this is what's more important in this crop. But I did like it's a good starting point. So what I came up with was scores for each of my crops in the end and their performances. So I'm gonna show you so that I don't bother you with all the math. So I had a toxin score because the tests I sent out revealed that my soil had toxins in them, had heavy metals in them. It used to be a parking lot. So maybe that's where they're pulling up the toxins. In the end, though, the toxins were like the same across the board, so they didn't make much of a difference. I used an NDS score that I created called a nutrient density score. And so for lettuce, Swiss chard, and kale, right now we're still waiting for the last of these. The kale and the lettuce, I'm regerminating again to get a score for this, but for Swiss chard it was 33% for germination as well for N-treated. So the control, which was just water, for lettuce, for Swiss chard, and kale, performed the best. So when you germinate your seeds, just use water. That's it, from my study. And then growth, the mixed results. We have mixed results. So the average weight and the yield of lettuce for the the control grew best for lettuce, where I had 20 crops at the end of my sowing season, and they weighed 1.4 versus the ACT, which was 1.23. The N-treated didn't yield as much lettuce, but they were the same weight as ACT. And then nutrient density, the ACT and the control had the same nutrient density. Just keep in mind that my control, when I filled up my beds, I filled it up with mushroom compost. This was how I was a farmer rather than a scientist. And then the scientist I was collaborating with said, you should have filled it up with just topsoil so that we could really see the effects. But no, I was afraid that I would lose my crops. So I filled it up with, the farmer in me filled it up with mushroom compost. So my control base is rich soil already. But, and so this is how, I don't know how statistically significant it is. It still bears doing more research for sure in more numbers of beds. But my control and my lettuce vied for highest nutrient density. And then in Swiss chard, we have the control having again the best germination, the control having the biggest growth, and the N treated as having the nutrient density, the highest nutrient density. So it's mixed results. So, and then kale, again, the ACT treated had the highest nutrient density, but, and the control for 50% germination, had 50% germination in kale, and the ACT treated, was the winner in kale. So it looks like these, ACT or compost tea affects different leafy greens differently. So it looks like it's very good for kale, but doesn't really matter so much in the others. I think, I don't know. So I'm still learning. That's why we have the second research. How do you get mushroom compost? We have, how did we get it? We had a truckload delivered 2 years ago when we started. And you can, if your source has mushroom compost as one of the things, not just topsoil or compost, then they can deliver it for you. So we happen to have a source for that at that time. I have to ask my husband, Dave, where he got it from. But yeah, so a little preview into what I'm doing next is I'm comparing the microbiome population induced by different microbial foods for the production of a safe and effective aerated compost tea. So in the first study, I found that these were the 3 families that I ended up with in my tea after brewing. I had increased their population from 0.8% in the vermicompost to 9% in my tea, from 10% to 36% in my tea, and so on. But I selected for basically these 3 families when I brewed my tea. Do we want to do this? Is this the permaculture way of doing things that we want to control the bacteria so much? Maybe for disease, if we know specifically what we are selecting for and what that particular bacteria does, and we have an issue with our plant that is such as the botrytis that is known to be where compost tea is known to be effective against botrytis because of the Pseudomonas, then maybe we want to do that. But I think for the most part, for the home gardener, you can just make your soluble compost tea from your bokashi or your vermicompost, and you'll be fine. Now, bokashi in particular, as you can tell, the majority of organisms found in vermicompost are potentially plant growth promoting. So this is another benefit of using vermicompost. I'm still working on identifying them all, so there is likely even more. This is from Dr. Jones, my collaborator. Now, bokashi, because it's anaerobic, then you have a different profile of bacteria there. And those, Dr. Jones, if you go to his website, Agrego Data, he has more studies on bokashi in particular, where he looks at the different bacteria in the bokashi vein, which is exciting. So that would be, if that's of interest to you, because that's your primary source of compost, then you know what you're getting when you do that. But also, going back again to that Korean study where you had the bokashi, or you had the different kinds of compost plus vermicompost, the more diverse your base of compost is to creating these teas, the better. So maybe you have a dusting or a watering of bokashi vermicompost one part of the year, and then you bokashi compost tea one part of the year, and then you have a vermicompost application, and you have some other parts. That would be probably the most ideal. The more diverse your inputs are, the better. What happens? So the second study, and I'm now 5 minutes in left, but what happens if we feed the compost different stuff? Do we grow different bacteria? So this is the question currently, and I've already know the answers to these guys. So we have alfalfa, kelp, humic acids, molasses, which they tell you never to feed because it's like sugar and they just die quickly. And so I will share that graph really quickly before I end. Escape. Yeah. Oops. Oh, that's the wrong thing to share. Ah, I keep on sharing the wrong thing. All right, Corona-bacteria for this graph. All right, so am I sharing the right thing? No. Let me see here. Okay, do you guys see the Corona-bacteria graph on the screen that says 0, 12, and 24? Yes, okay. So this is where it's interesting, and maybe it's not best seen on a chronograph, it's better seen like on a different 2D. But this is again the vermicompost that we start with at time 0, at hour 0, and at 12 hours you see now which, oh dear, that this doesn't tell me what feed I put in it. Okay. All right. So I have to like, okay, not this one, fine. We'll go back to this one. This is a better way to see it. Present. Okay, so if you look at it this way, I have these different letters to represent different things here. Control, AM is alfalfa meal, KM is kelp meal, humic acid, molasses, fish hydrolysate, and they're at 24. So these are the 24-hour results, and these are the 12-hour results here. What do the results tell us? And then this is the diversity in color. So each color is a different phyla. So Proteobacteria, Firmicutes, Bacteriodota, Actinobacteriota, etc. Each color is a different phyla. So look and see which line is the most colorful, and that will tell you which is the most diverse. So I have my arrows on the control at 12, and 24 AM. Is that the right one? 12 and 24. Okay. 12 and 24. 12 hours and 24. Was that really the first one? Control, humic acid. Okay, humic acid 12. So the control, okay, so yes, the control is the most diverse because we didn't do anything. We didn't feed anything. So contrary to what Dr. Elaine Ingham had said about if you don't feed it, then all the bacteria would die. It actually, this particular test showed that after 24 hours, if you didn't feed it anything, your original was similar to the 12-hour, and the 24-hour were the same. Okay, the next most diverse bar went to the award. Most diverse bar award goes to humic acids. So another thing that we noticed was that whenever you put humic acid, if you want to feed your bacteria, whenever you put humic acid into the brew, you buffer the diversity. So, here are the results on the right. The vermicompost has no detected pathogens. There's nothing in the tea that wasn't first there in the compost. If we don't feed the tea anything, the bacteria are still alive and diverse after 24 hours. Molasses, fish hydrolysate, and kelp all tend to decrease bacterial diversity after 24 hours. Different feeds select for an abundance of different bacteria, and humic acids tend to keep diversity. So, That's just, again, the finding, the same thing. Vermicompost is a suitable base compost used for compost tea. Without humic acids, aerated compost tea becomes less diverse. Around 3 bacterial families dominated after the bubbling. Oops. Molasses makes the bacteria spike and die faster. Bacteria diversity is still in fact intact after bubbling for 24 hours unfed compost extract. Why? Maybe it has innate humic acids. I don't know why. Maybe my particular vermicompost had humic acids in it already from just the organic matter that was there. I don't know why. So, to answer that question again, I would say the first answer is ACT a viable fertilizer like to nitrogen? I think in the case of kale, yes, but in lettuce and Swiss chard, not so much. Does ACT make leafy greens go bigger? Not necessarily so, I think, just in the case of kale. And then does it become more nutrient dense just in kale? So in lettuce, ACT improved nutrient density and Brix, but not root development. Kale, ACT boosted nutrient density and resilience. Swiss chard, no yield benefit. Nitrogen improved. Actually, nitrogen was better for Swiss chard. Sustainability, ACT is a viable organic alternative for some crops. And microbial selectivity, ACT selects for a few microbes. They may drive specific crop responses. So do you all want to know compost tea? How to make compost tea? It's at the top of the hour. Or are you guys all good? I'm just going to leave you, because it is the top of the hour, with this podcast by Compost Tea Master Troy Hinkey. If you want to just screenshot this or copy the bit.ly link, he has a podcast on how exactly to do this right. And he sends his vermicomposts to Zach as well, to Dr. Jones as well. So I know he has good stuff that he makes. So I hope you got that bit.ly/Troy-Hinkey-compost-course. That's also on our website, permaculturegardens.org. But in short, you bubble it for you don't have to bubble it, as you know. This is how I did it. This is the brew that I used. This is like a recipe that I found online, but in the end, what I did is I didn't use molasses, I just used humic acids. And I use humic acids and fish hydrolysate now when I brew the compost tea, always with humic acids. And then I have a video of Dave making nettle
Hi,
tea.
this is Dave from Permaculture Gardens. We do a lot of experimenting, and sometimes these things don't work out, but we thought we'd start sharing with you the experiments as we're doing them, just in case they do turn out to be useful, and it just guides you through kind of the things that we're experimenting with. Here we're trying to make a non-aerated compost tea. So I've gathered about 1 kilogram or 2 pounds of a mix of comfrey and stinging nettle. And this is supposed to be a really good mid-season drench or nutrient feed for things like tomatoes, peppers, your fruiting crops in the summer that need that extra boost. So I've gathered all this. I've already started chopping it up. So all I've done is I'm going to wrap it up. I have here some Agribon row cover. It's like a breathable plastic row cover that I use in the winter for protecting my low tunnels. But I'm repurposing it because if you have something like an old stocking that's supposed to also, like a hose, you could also fill all the materials in there. You have to be a little careful with the stinging nettle. It actually will sting you. That's why I'm wearing gloves. The reason why I'm chopping this up instead of just wrapping it all up is that you want to increase the surface area so we can extract as many of the nutrients out of these plants as possible. So after I cut all of this up, I'm going to wrap it up like a bag, and then we're just going to put it in a non-chlorinated water for about a week. We'll stir it every day. It's non-aerated, so I'm not using a pump. So this is probably one of the easiest compost teas that you can make. Now, this is the first time I've ever made this, but once I've made it, we're going to test it on some of our tomato plants, and see if it boosts growth, and then we'll report the results to our tribe. Okay, so I can cut this up. Get all of it in there, and then I'm gonna wrap it up.
You want my stockings?
Well, you don't have old stockings, right?
Well, there's I have stockings. Yeah, I do.
Well, I've already got this all set up. Go with this for now. Because I think if you already have some agribon, it's probably easier just to repurpose that. Okay, so we got it. So now we'll just soak it in here.
It's just cold water.
It's okay, so it's just cold water that I got out of a rain barrel. If you're gonna get it out of your tap, let it sit for 24 hours so the chlorine can dissipate before you put the plant materials in. That's all you have to do. So we're gonna let this soak for the next week, and then we'll use it as a foliar spray and a root drip for our tomato plants and our peppers. Thanks.
All right, and that's it. So that's the end. Do you have any questions?
If I make a compost tea with just like 3 spoons of, the vermi compost, like in a watering can, how long should I leave it in the water before I water the plants with it?
Well, from my experiment, it said like you can leave it for a day, right? And it'll still
Yeah.
sustain diversity. Yeah,
Okay. So if I just leave it like overnight or something and then
you'll be fine.
it'll be
Yeah.
fine. Okay. Great. Thank you.
You're welcome. I hope this has made it simpler for us to just not be scared, or daunted by like this concept of compost tea and am I doing it right? But just like go ahead and do it with whatever you have already, whether that's bokashi and or vermicompost or just compost old, that's outside, that's decomposed. Yeah. All right. Susan, how would I use bokashi for compost tea? So you have the dregs of the bokashi water. Just add that to your watering can and then water. Sometimes this is not just me, but like other Asian growers that I've met, they say when they wash their rice water, they don't wash the water down the sink. They save the water from the rice and then they water that they water that in the plants because it's a sort of ferment. The rice ferments things around it really easily, ferments really easily, decomposes really easily, and that produces, a specific kind of bacteria that's healthy for growing. Yeah. Oh my gosh, thank you, Dr. Westerveld. Oh my gosh, we've been doing college tours recently, and every time I go, I'm so excited because I feel like I want to go back to school again, and I think about you and your perpetual love of learning. Um, but yeah, Dave is the opposite. He's like, I do not like universities. He's the one with a master's, but he does not like any like formal college thing. Okay, thank you. Everybody asks us. So the nettle tea worked out well. Like we had good non-blossom rotty tomatoes then that year, and I think that for tomato, that's why I'm studying tomatoes in the second. That's the practical application of that second research, tomatoes in particular, because I'm wanting to know, does it really make a difference, right, when we use the compost tea for tomatoes? Because that's the important one. Oh, thank you. Yes, I've been looking at I've been looking at courses. But yeah, one thing at a time. Yes, thank you. Thank you for the vote of confidence. Well, yeah, so this was quite a pleasure. Thank you for hearing me out and being curious to know what the deal is with compost tea. And I hope that if you have any questions after this, the resources are all online in my compost tea website, and I keep on updating them. Just shoot me an email and I'm happy to answer. Thank you. Oh, thank you, Susan. Thank you, everyone, for joining. I'll see you again in the next time. Bye.
Thank you. Bye.